Welcome, everyone. Thank you for joining us for today's webinar, Overcoming Limitations of Conventional Tag Systems, Strep-Tactin XT Applications. It is presented by Dennis Karthaus, scientist and team leader for cell cultivation at IBA Life Sciences. My name is Judy O'Rourke, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is a leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. Simply click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box. Our speaker will respond to your questions via email following the presentation. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Now, without further ado, please join me in welcoming Dennis Karthaus. I will now turn the presentation over to him.
Thank you very much for the introduction, and I'd like to thank LabRoots for inviting me to this great conference event. So when you use tag systems, you can use them for two applications. You can use them for the purification of proteins, or you can use them for immobilization of proteins on surfaces like ELISA plates or SPR chips in order to perform assays. However, as long as you want to use the tag system for both applications, you have to find a compromise. Because for immobilization applications, you want to have a binding affinity as high as possible in order to have a stable immobilization of your protein of interest. On the other side, when you look to purification, and especially if you want to purify your protein under physiological native conditions, binding affinity that would be so high will make it impossible to elute your protein efficiently from the column. So therefore, you have to find a compromise which allows you to elute your protein from the column, but on the other side, immobilize the protein on chips. And yeah, that's a kind of impossibility. However, our Strep-Tactin XT system can overcome these limitations. And in the talk today, I would like to give you some application examples of how you can use the Twin-Strep-tag Strep-Tactin XT system in order to do both purifying proteins and use the system for immobilization of proteins. The talk is divided into two sections. In the first part, we will look to the purification of Twin-Strep-tag proteins from Expi and Expi293 media. And in the second part, I will give you some applications of how you can use the system for SPR analysis or for the use in the switchSENSE technology from dynamic biosensors. So first, let's have a brief look to some existing tags on the market. One group of tags are so-called epitope tags, like the flag tag or the MIG tag. These tags usually provide high protein purification or high purity, but the resin capacity of these tags is very limited and the capacity is low, so purification using these tag systems are quite expensive. In addition, in order to elute your protein, sometimes it's necessary to change the buffer conditions to harsh buffer conditions, like a low pH value, and that can degrade your protein. Another group of tags are large tags, like the maltose binding protein, the GST tag, or the strep tag. These tags can increase the solubility or the expression of your protein of interest, but on the other side, they can also have an impact on other properties of your protein, and that's something you want to avoid. In addition, due to their large size, they are highly antigenic. And because of this fact, you have to cleave off the tag before you work with your protein of interest in animals. And last but not least, you can use these tags not for purification under denaturing conditions, because if you do this, you would denature your tag as well. So the most widespread tag system on the market is the HIS tag system, because it's a cheap system and the binding capacity of the resins is high. However, very often the purity of his-tag purificated proteins is not high enough in order to do your following experiment, and then you have to do another purification step. In addition, you often have to empirically determine which resin is best for your application, for example, nickel anti-A or talon. And you have to play around with the imidazole concentrations in the sample buffer or in the watch buffer in order to find a compromise between yield and purity. In addition, the system has a binding affinity in a nanomolar range, and that makes it not very well suited for SPR applications because you will measure a significant dissociation of the tag itself. And finally, when you purify samples from mammalian hosts or insect hosts, you can co-purify many host cell proteins because these hosts have more host cell proteins with histidine residues. So let's have a brief look to the Twin-Strep-tag Strep-Tactin XT system and how the purification with the system works. In the first step on the right corner, you can see the loading step. You can load samples from bacterial lysates, cell lysates, yeast lysates or insect lysates, or supernatants from insect cells or mammalian cell culture. You directly load your sample on the Strep-Tactin XT column, and your protein of interest, which is fused to the Twin-Strep-tag, will be bound on Strep-Tactin XT by binding the Strep-tag in the biotin binding pocket of Strep-Tactin XT, which is a re-engineered streptavidin. Once you've bound the protein of interest on the matrix, you can wash away host cell proteins. You can also work very efficiently under physiological conditions, but you can also use high detergent concentrations, high salt concentrations, etc. And then it comes to the elution, and for elution you only have to add biotin to your wash buffer in order to elute your protein. Because what happens, each of the two tags from the Twin-Strep-tag has an affinity to Strep-Tactin XT in a nanomolar range. So from time to time, one of the two tags will dissociate from the binding pocket. However, the protein is kept in place by the second tag. But once you add biotin to the buffer, the biotin will block the binding pocket. And then after the second tag dissociates, this pocket will be blocked too, and that elutes the protein under very gentle conditions, and you can work effectively with physiological conditions in the system. Finally, you can regenerate the column by just adding NaOH and go for another purification round. And we have two purity conferring steps in this protocol. First, the specific binding of the host of the protein of interest using the Twin-Strep-tag, and the elution itself, because you don't change any buffer conditions, you only add biotin, and proteins that might be bound unspecifically to the resin will not co-elute with your protein of interest. So now I want to show you some data from our study purifying Twin-Strep-tag proteins from Expi supernatants. And around 1 year ago, I talked to Henry Chu from Thermo, and he told me that his tag users are often having problems purifying their proteins from the Expi media because during the loading step, protein leaks from the column. And in order to have an efficient his tag purification, it might be necessary to add a dialyzer step or to add reagents to your supernatant. And we were wondering if we will face the same problem when we use our Twin-Strep-tag Strep-Tactin XT system. So here's an experiment shows what happens if you purify a his-tag protein without any adjustments of treatments of the supernatant on a nickel anti-A column. Here we spiked sherry protein fused to a 6 histidine tag to Expi supernatant, and we loaded 20 milliliter of the supernatant on a 1 milliliter nickel anti-A column. And you can see that during the loading step, protein is leaked from the column indicated by the white bright area in the upper part of the column. So the first question is our maximum binding capacity influenced by substances in Expi media? And for this experiment, we looked to the maximum binding capacity of our Strep-Tactin XT columns. For this experiment, we spiked Expi and Expi-293 media with 3 different proteins, and sherry, a red monoclonal antibody, and secreted alkaline phosphatase, all fused to the Twin-Strep-tag. And we adjusted the concentrations in the supernatant to two concentrations, 100 micrograms per milliliter or 200 micrograms per milliliter. And all experiments were done on 200 microliter Strep-Tactin XT gravity flow columns. This is important because in gravity flow mode, we cannot adjust the flow speed. So if you look to the binding capacity, which was determined by measuring the amount of eluted protein after the columns were overloaded and washed, you can see that for M-Sherry and the phosphatase, the protein concentration, the supernatant, had no significant impact on the maximum binding capacity for both Expi and Expi media. However, when we look to the monoclonal antibody, we can see that the binding capacity was affected and decreased for the lower concentration. But this is more a general chromatography problem than an issue with the media, because the monoclonal antibody is larger than M-Sherry and secreted alkaline phosphatase. It is 5 times larger than M-Sherry and 2 times larger than the phosphatase. The nanomolar or the molar concentration of the antibody in the supernatant was lower, and as it's larger, it needs more time to diffuse into the pores. So we have a lower diffusion force into the pores, and due to the very low residence time of only 20 seconds, this residence time was not enough to allow the monoclonal antibody to enter completely the pores and make use of all available Strep-Tactin XT ligands. The low residence time is a result of the use of the gravity flow column. As we cannot adjust the flow speed here, the residence time was so short. However, in order to verify this, we did another experiment with the Expi supernatant. And in this experiment, we loaded the supernatant on the column using an automated pipetting device, which allows us to control the flow speed and adjust the residence time to a residence time of 2 minutes. And you can see that by doing this, we increase the binding capacity for all 3 proteins. And very important is here the theoretic M-Sherry capacity that you can see with this light gray line on the right side of the graph. And this is a capacity which we expect, or which is a theoretic maximum M-Sherry capacity for Strep-Tactin XT column based on the amount of coupled Strep-Tactin XT ligands. And you can see if we increase the residence time, we achieve this maximum binding capacity, and that verifies that there are no interfering substances in the media that leads to leakage of protein during the loading step, what we have seen for the his-tag purification at the beginning. Another question is, how high is our recovery and purity? And in this experiment, again, we spiked Expi and Expi supernatants with our 3 proteins at our 2 concentrations. But in this case, we only loaded 75% of the maximum column capacity on the column. Again, we washed away impurities and then we eluted the protein. And you can see that for the M-Sherry and the phosphatase, we get high recovery rates in the range between 90 and 100%. And for the monoclonal antibody, it was a little bit lower, but again, due to the low residence time of 20 seconds, this leads to a little bit lower recovery compared to the smaller proteins. And by increasing the residence time, you can overcome this problem. The data for the Expi experiment were quite the same. That's why we only show here the Expi results. And instead of the Expi bars, you can see on the right side the purity of the purified proteins in the SDS page-GL analysis. And you can see that all proteins were purified at very high purity, close to 100%. For the M-Sherry protein, you can see 2 bands in the SDS page, but the second is known as a degradation product of M-Sherry that you will always find, so it's not a real contamination or impurity. And you can see, when you look to the secreted alkaline phosphatase, that protein concentration had no impact on purity. In another experiment, we wanted to see what happens if we purify proteins from Expi supernatants at very, very low concentrations. The Expi systems provide very high titers, but if you have a very hard to express protein, even these powerful systems can result in a low concentration of protein in supernatant. And in this experiment, we spiked Expi supernatant with our 3 proteins, but only at a maximum concentration of 20 micrograms per milliliter. And if you look to the recovery, you can see that we were able to efficiently purify all 3 proteins from the Expi supernatant. And in this experiment, we purified 50 milliliter of Expi supernatant over a 300 microliter Strep-Tactin XT column. On the right side, you can see the SDS page analysis of the experiment, and you can see that we have high purity for all 3 proteins. However, you can also see 2 contaminating bands in every elution, and the band profile looks quite similar to the profile of an antibody. And actually, the provided supernatants from Thermo were from Expi cells who already overexpressed a human antibody at a very high titer, around 3 grams per liter. So this is a kind of artificial contamination, which is usually not present in the Expi supernatant. So in order to verify this, we plotted the supernatant, you can see this on the left side of the slide, and the elution fraction from the secreted alkaline phosphatase elution. And after plotting, we detected the heavy chain of a human antibody by an anti-human HRP conjugated antibody. And you can see that we were able to detect the heavy chain of this overexpressed antibody, of course, in the supernatant, but also in the elution, which verifies that this contamination is a kind of artificial contamination. However, it gave us a great opportunity to challenge our system, because we wanted to see, okay, how we can deal with even this challenging purifications where we might have high concentrated contaminations. And then we made use of a benefit of the system, because the Twin-Strep-tag Strep-Tactin XT system has a binding affinity in a picomolecular range. So this is the highest binding affinity available among all commercial available tag systems on the market. But as it relies on avidity, as I've shown before, it's still reversible. But this high affinity now gives us a possibility to perform very intensive wash procedures in order to remove such contaminations without losing our protein of interest. And here we shoot a little bit with Canon on birds, but we washed the columns with 30 column volumes of wash buffer after loading the supernatant. And you can see in the SDS page here on the right side that this makes it possible to remove the high overexpressed antibody completely from the elution fraction. And I think this experiment shows you quite nicely how you can use the system for even very challenging purification applications. So finally, we did an experiment where we transfected Expi-293 cells with secreted alkaline phosphatase vectors or a red monoclonal antibody coding vector, expressed the 2 proteins for 4 days, and then we purified the supernatant over a Strep-Tactin XT high capacity column. And first you can see the yields we achieved with the system was 300 milligrams per liter for the secreted alkaline phosphatase and 86 milligrams per liter for the red monoclonal antibody without any optimization here. And if you look to the purification, you can see that, again, we were able to achieve very high purities for all 3 proteins. And I think these data shows you that there are no interfering substances in the Expi or Expi-293 media. So the systems work quite well with our Strep-Tactin XT system, and it provides high recovery and purity rates. Finally, a picture is worth 1,000 words. On the left side, you can see the column from the beginning, from the histec purification. And on the right side, you can see what happens if you directly load Expi supernatant spike with the Twin-Strep-tag and Sherry on the Strep-Tactin XT high capacity column. And you can see that here we have a very nice binding in the upper part of the column without any leakage of protein during the loading step. So After looking to the purification, now we want to see how Strep-Tactin XT and the Twin-Strep-tag can be used for immobilization applications and assays like for SPR. Because if you have a binding affinity in a picomolecular range, it makes totally sense to use the system for immobilizing proteins. First, let's have a look to some advantages of the Strep-Tactin XT system for capturing your ligand instead of directly couple your protein of interest on the chip. First, the ligand is not inactivated or influenced by the immobilization procedure itself, because you use the Strep-Tactin XT Twin-Strep-tag for immobilization. You can do this under native conditions, under physiological conditions, and can choose a buffer condition for immobilization that works perfect with your protein of interest. In addition, you don't have to modify your ligand anymore, for example, doing a biotinylation step. And you have high affinities, which enables you to do long-time measurements and measure very high binding affinities between the two binding partners. The chip can be regenerated easily by just regenerating the Strep-Tactin XT Twin-Strep-tag binding pocket. And finally, you don't have to have your ligand highly purified in order to immobilize it, because you specifically capture your ligand out of any sample using Strep-Tactin XT and the Twin-Strep-tag. So the following data were provided by Biofin, and in this first test we wanted to see how reproducible we can immobilize Strep-Tactin XT on an SPR chip. And in this experiment, 4 CM5 BioProcessing chips were coupled, or Strep-Tactin XT was coupled to 4 CM5 BioProcessing chips. And you can see from the results below that the signals that were obtained for any chip after each coupling were very reproducible, and you can see it also from the curves that the coupling procedure was highly reproducible. In the next step, we captured 3 different Twin-Strep-tag proteins, and Sherry, again the monoclonal antibody, and GAPDH on the chips that were coated with the Strep-Tactin XT. So what we did is we captured the protein by loading the protein on the chip, and then after this we regenerated the chip by 3 injections using 3 molar of guanidinium hydrochloride. And each bar you can see in the graphs represent 1 capture step, and in the 3 injections 1 regeneration step. So in this experiment we did 12 capturings and regenerations in total. And you can see that the signals for each capturing step for all 3 proteins are very reproducible with a CV value below 1%, and that we were able to regenerate the chip completely after the 3rd injection. And therefore, you can use this chip many times and get a high reproducible measurement of your bound protein. We also tested another kind of chip, the CMD200M chip from Xantec, and these studies were done by the University of Applied Sciences in Bielefeld. And here we coupled an anti-CD4 FAB fused to a Twin-Strep-tag on a Strep-Tactin XT coated Xantec chip. And again, you can see that we get very reproducible signals in the capturing step, and also a completely regeneration of the chip using guanidinium hydrochloride. So these data were provided by Alexander Yeliseev, and here you can see the coupling of a GPCR protein on a Strep-Tactin XT coated chip by using the Twin-Strep-tag. And you can see that we have here a very stable signal over a long measurement time, which verifies this very high binding affinity of the Twin-Strep-tag to Strep-Tactin XT. And it also shows you that the system works very well with detergent and immobilization of membrane proteins. SPR is one way how you can measure binding affinities and kinetics, but there are also existing alternatives, and one very interesting alternative is the switchSENSE technology that can be also used for kinetic studies and measuring binding affinities, but also for many other applications. And this technology is provided by Dynamic Biosensors, and they provided the following data, which are from their development of a Strep-Tactin XT chip. First, a brief look to how the system works. In the switchSENSE technology, you have a double-stranded DNA, and one strand is coupled to a fluorescence dye, and the other strand is coupled to your analyte, or in this case, to Strep-Tactin XT. So once you apply a current on the chip surface, the DNA will be pulled down to the chip surface, and there the fluorescence signal is quenched. So in the moment where you now bind the protein to the Strep-Tactin XT, or then another protein to the already immobilized ligand, you will increase the friction coefficient due to the larger size of the whole complex. And this will reduce the movement speed when the double-stranded DNA is pulled down to the chip, and therefore influence the quenching signal, and that gives you a dynamic response that you can see on the left side. So the larger the protein is, or the more protein is bound to the chip, the higher is this signal. You can use this technology for binding studies like measuring KD on, KD off, or KD values, or avidity effects. You can also use it for measuring strokes, diameters, measuring the amount of dimers, trimers, or aggregates in your samples, or configuration changes of your protein. And furthermore, you can do stability studies with this technology. So in this experiment, at the upper part of the presentation, you can see the association and dissociation rate of a Twin-Strep-tag GFP that was coupled on a Strep-Tactin XT functionalized switchSENSE chip. And you can see that we have high association rates and that we have a very low dissociation of the sample. And the KD value, which was determined with this technology here, was 64 picomolar, so very, very high affinity. And in contrast, when you measure the binding affinity of the his-tag nickel anti-A binding with the same system, you achieve a binding affinity in the range of 2 nanomolar. So we have a significant higher binding affinity for our system. In the experiment below, you can see the binding of a Twin-Strep-tag protein A on the Strep-Tactin XT switchSENSE chip. And after that, an antibody was coupled to protein A. The antibody was adenomab, and this binding is very well characterized in the literature. So it was possible to compare the values we achieved with the Strep-Tactin XT chip to the literature values, and they agreed very well, which shows you that you can use the Strep-Tactin XT chip instead of a covalent coupling of your protein, of your ligand, in order to capture finally the protein that you're interested in, or to measure binding kinetics. Another thing you can do with the technology is shown in this slide. Here, Dynamic Biosensor uses strep-avidin because this study was done before the Strep-Tactin XT chip was available, but the same study can be performed with Strep-Tactin XT chips. So you can see that once strep-avidin was bound to the double-stranded DNA, the effective diameter has a specific value of around about 6.5 nanometers. Once you bind now a biotinylated protein, the effective diameter increased to 7.5 nanometers. And then if you have a conformational change of the protein to an open configuration, you have a further increase in the effective diameter, which makes it possible to measure the distribution between open and closed conformations. The advantage of using a Strep-Tactin XT chip is that you don't have to biotinylate your protein. You can use the same tags that you have already used for purification, but you still have a very high binding affinity. And that brings me to the summary of this presentation. The Strep-Tactin XT Twin-Strep-tag system is a universal system as it allows efficient purification of proteins and immobilization of proteins. We have a high binding affinity in a picomolecular range, but we still have a reversible binding that makes it possible to use the system for purification as well. In addition, it's effective in immobilization of twin-strep proteins on SPR chips or the switchSENSE technology, but we can also easily purify proteins at high purity from mammalian systems, and it's quite compatible with the Expi system, providing purities of more than 98% in most cases. And finally, the tag is very short. So the Strep-Tag has 8 amino acids, and the Twin-Strep-tag consists of 28 amino acids. So for most applications, it's not necessary to cleave off the tag, and the tag proteins were efficiently used in animal studies from our customers. With that, I thank you for your attention, and if you have any additional questions, please do not hesitate to contact me under the given contact details, and have a look to our website for further information about our products. Thank you very much.
Thank you, Dennis, for that informative presentation. I'd also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that this webcast will be available for on-demand viewing through April 30th, 2019. As a final reminder, our speaker will follow up with any questions you've submitted via email. That's all for now. Thank you for joining us, and we will see you next time. Goodbye.